This is Professor R.M. Mahendrakar from JSS Arts, Science and Commerce College, Gokak. Today we'll be going to discuss about the plant tissue culture. Now this plant tissue culture, we are going to study it in three parts. In the first part, I'll be telling you about the brief history of tissue culture and scope of tissue culture. In part 2, we'll be studying about the applications of tissue culture and morphogenesis. And in part 3, we'll be discussing about the technique involved in the plant tissue culture. Agriculture today is on the verge of technological revolution in a manner it has never been seen before. As we enter the new millennium, one key development that comes to our mind is the emergence of biotechnology, which offers some of the best opportunities and solutions to some of the uncontrolled problems faced by us. Biotechnology is a group of technology that shares two things in common. They manipulate the living cells and the molecules, and they have a wide range of uses that can prove and improve our life. The major techniques of biotechnology are the genetic engineering, cell culture, tissue culture, bioprocessing, protein engineering, etc. So in this presentation, we'll be studying about the brief history of plant tissue culture and scope of plant tissue culture. Plant tissue culture or cell culture or micropropagation is the technique of producing desirable agricultural quality plants in large number from a small piece of plant in a relatively short period of time. Through this method, entire population with premium qualities can be created from a single elite specimen. German botanist Gottlieb Haberlin proposed the importance of plant tissue culture and cell culture in isolation for the first time in the year 1902, and he is regarded as the father of the plant tissue culture. He used the tissues of Lamium, Ecornia, Ornithoallium, Musselinea for his experiment. Later, von Oberach in the year 1941 demonstrated for the first time that when coconut milk was added in the nutrient medium, it produced callus in the thura. This proved a turning point in the field of embryo culture. Subsequent detailed work by Raghavan and Torre and others led to the development of synthetic media for the culture of younger embryos. Gautheret and White successfully cultured cells of Salix, Nicotiana hybrid, and carrot on synthetic media. They for the first time demonstrated that growth regulators and vitamins, if added to media, enhance the growth formation of cells and resulted in formation of the callus. Skoog and Miller demonstrated the introduction of division in isolated, mature and differentiated cells by using synthetic as well as natural compounds. Guha and Maheshwari demonstrated the possibility of raising a large number of plantlets from pollen grains of the thura. In 1972, Carlson and others produced the first somatic hybrid between two plants by fusing their protoplast. The basic process in tissue culture is as follows. Depending upon the species in question, the original tissue piece is taken from a shoot tip, leaf, lateral bud, stem or root tissue of the mother plant. Here in the image, we see we have taken a piece of the stem of this plant. This is called explant. These explants are first sterilized and then they go through the initiation, multiplication, and rooting methods for producing a cell into a full-fledged plant. These ready plantlets are then hardened in climatically controlled greenhouses or polyhouses. Depending upon the species, the plants become ready for the plantation in the fields. Now we shall see about the scope of plant tissue culture. The first scope of plant tissue culture is mass multiplication of elite clones. Micropropagation allows the production of large number of plants from small pieces of mother plant. The production requires relatively short period of time to grow plants. Depending upon the species under production, a single explant can be multiplied into several thousand plants in less than 1 year. Elimination of diseases in planting materials. Another purpose for which plant tissue culture is uniquely suited is obtaining, maintaining, and mass propagation of specific disease-free plants. The concept behind indexing plants free of pest is closely allied to the concept of using tissue culture as a selection system. Tissue known to be free of diseases under consideration are physically selected as the explant for the plant tissue culture and they are produced in large numbers. Next, plant improvement through tissue cultures. Creation of superior variety of agricultural crops is possible only through tissue culture methods. Now in this image you can see one variety of this okra, that is the lace finger plant. Here the fruit production is very less and the same is somehow manipulated by the genetic recombinations, and here you are getting a plant which is showing a high yield of these okra fruits, and this variety now using the tissue culture methods can be produced in large numbers. Thus tissue culture plays a very important role in plant improvement. The next is the true to type production. That is, through tissue culture, you can produce the same clones of the plants in large number. Now here you can see this is a red okra plant, and this red okra plants can be produced in large numbers by taking a small piece of the tissue and producing a large number of the plants, and all those plants will be showing the same characters, the same physiological characters, the same anatomical characters, and the same phenotypic characters. Hence, through tissue culture, you can obtain true to type production. The next scope of the plant tissue culture is higher yields. Tissue culture plants have increased branching and flowering, greater vigor and higher yield because the explants are selected from the varieties that show all the better characters. Plant tissue culture is beneficial when conventional propagation is difficult. Conventional propagation means the normal mode of reproduction. Now, there are so many varieties of plants where the normal method of propagation through seeds and vegetative methods is not very easy. It is difficult. So in these cases, beneficial varieties can be propagated in large number only through tissue culture. Examples: rare orchids, rare flowering plants, etc. The next is the efficient method in saving space and energy. Now, through tissue culture, you can produce a large number of plants in a small area. For example, in a conventional method, the plants are grown in open form requiring an area about 25,000 square meters. The same number of plants would require only 10 square meter space if they are grown in tissue culture laboratory. Thus, tissue culture helps in efficient method in saving space and energy. Flexible method. The flexibility of nurseries can be improved as the capital investment of mother plant is reduced to almost zero. It may be easier to adapt to the changing conditions. Now, innovative methods. That is, through tissue culture, you can produce numerous number of new varieties. Now here in the image you can see I have shown you some 9 varieties of hibiscus flowers. Actually, through tissue culture at present, there are more than 54 varieties of hibiscus showing the different variations in their flowering color. So thus, tissue culture plays an important role in the plant culture. So with this, I conclude this topic on the tissue culture. That is the first part of tissue culture, where we have studied the brief history of tissue culture and the scope of tissue culture. Now in the part 2, we'll be discussing about the different basic terminologies used in tissue culture, the different types of applications of tissue culture, and also morphogenesis. Thank you. Take care and be safe.